looks like it's live. Doing some audio checks here real quick. Test, test, test. Test 1 2 1 2. That's weird. I can't hear myself. Can't hear myself on the loop back thingy. I think it's too low. Let me adjust it. Oh yeah, I think it was too low. Let's see if I can get JX3D's music going here. Out. I think I got it now. Let's wait for the pass through. There's always a delay. Sorry, a bit rusty. I haven't done this for at least 6 months, so I had to reset a bunch of stuff up in OBS. Okay, so I think we're okay. Music is still playing. View meter looks okay on both. I'm just gonna go with it. So let me set it up so I can see the chat a little bit better. Okay. Sorry for all the setup crap. Took a while to get sorted this time. But anyway, here we are with the dark field image of the water sample. I'll show you where I got the water sample. My favorite spot where I used to work part-time, Palo Baylands Nature Center. The last time I actually worked there, back in 2014, 2015, whatever it was, for about a year or so, along with the zoo and some other stuff I was doing there. down the street. Gave me the idea for this channel, so it makes sense to keep going back to the well, and it's also an incredible spot for samples of all kinds of plants and microorganisms, and great for bird watching. And if you're into airplanes, there's a local airport nearby. You can watch planes take off and land. So a little something for everybody. There's also a little nature center out there. It's pretty cool. They have private parties out there once in a while at night, and they have restrooms for the public when they're actually open. Unfortunately, the building's only open on weekends now, which is a real bummer because it's an incredible resource out there. When I used to work out there, it was weekends and weekdays, certain weekdays, and now it's weekends only, which is very sad, but that's a whole political city thing I won't get into. But whoever's running it now is crazy because it could be so much better. So let's see what we got here. I want to show you where I got the stuff from. So I believe, there you go. So this is site number 1 where I got some stuff. It's right by the main gate where you walk in on the levee, the big dirt levee to get into the facility. And as you can see, there's a bunch of pickleweed here. There's that pool area. So I actually kind of walk down. It's very easy to sink into the mud here unless you're wearing boots. And I also don't want to, it is a nature preserve. And the whole point is to preserve the area and not put a bunch of stupid footprints and mess up the area. So the goal is to get in, minimal damage, get your water sample, get some plant samples, and get out. And so I just pretty much right where that the first shadow of the log fence is going across the picture on the bottom. That's where I walked across to this little edge area, grabbed some water samples there, and that's what we're looking at here in the scope, first and foremost. And there will be more samples to come. So you can see it's mostly pickleweed here. There'll be some other things mixed throughout here, but since this is a saltwater habitat, you're not going to find too many plants here. You'll find pickleweed, salt grass on the edges, cordgrass, and I'll show you what's cool about cordgrass later. But there are certain plants adapted to live in salt water and have mechanisms to deal with how they deal with the salt without killing the plant. So that is the place I'm doing it at. So now let's go look around here a little bit for some stuff. There's some debris here. I'm on dark field right now. You can see some flagellates jumping through here. Tell by that kind of wavy pattern where they don't go in a straight line and they don't spin on a dime. They actually go through wavy bits. Oh, nice. Look at that. That's one of my favorites right there. That is a, so if I can zoom in on the digital zoom a little bit to make it easier. That is a little Stylonkia with the nice little, see, it's the cilia are pumping and making all the little particles in the background dance around. You get a little visitor there too, coming to it. But they have those at the poles, I would call it, the top and bottom of they kind of have a bilateral symmetry a little bit compared to some other animals that are just circular, radial symmetry. And anyway, so they have these poles, and the cilia are fused into these thicker structures called cirri, C-I-R-R-I, at the poles there. That's so cool to see that with the particles moving around. Trying to get a little bit sharper there. It's a little having trouble with the sharpness here. I have to boost that light a little bit more. But the dark field is great because you can see all the particles in the background cycling around as the vortex is created by the pumping cirri and other cilia on the ciliate. Really cool. Let's see if I can go a little bit closer before it leaves. Now we're at back to bright field again. There you go. There's a better shot of the little Starlonkia do before he takes off. And you can see the cilia pumping away there. So the cirri are now kind of left and right on the image. And you can see as I focus through, you can see that they're all over the edges as well, but mostly concentrated at the poles. I would call it what I would call the poles, the top and bottom of the tapered part of the cell. But as I focus, they look like little thorns almost sticking out. They're very thick cirri. So very cool. I like these guys a lot. Okay, let me move this over a bit so I can see the chat. Right now I'm blocking my chat. No chatties yet. No problem. This is a weird time. Anyway, so that's one of my little favorite guys there. There's one that's similar to this that actually walks on the cirri, and it's kind of a concave shape. It's more of a disc shape and concave. It looks similar to this one, but this one's not quite as small and not moving the same way, but they can actually walk on the cirri like legs. So it looks like an insect walking on top of another thing. There's a diatom coming in for view. On a different focal plane there. Some better light on that guy. Diatom going off to the side. But yeah, I love these guys. They're really cool. But there's one is similar to this guy, Euplotes, which is a little bit smaller and behaves a little differently. It does look a lot like this guy, though. I don't think it's a Euplotes. I haven't seen Euplotes for a while, but they have this kind of, you see the little structure on the right, almost like a comb. I don't know if I can point it out on the, I don't know if my actual cursor will show up here. Let's see. Well, my cursor, yeah, the cursor does show up there. So there's like a little comb structure right there, where the cilia are over kind of like this oral groove thingy. That's where they take in the nutrients and form vesicles to bring in and take out stuff. Since they don't have a proper mouth, it's just a single cell, remember? But they have pretty complicated structures for something that's a single cell. But that structure right there is like kind of where the oral groove is. And that's part of why they're beating cilia so much. Is it sitting in place, doing kind of bringing food to them. And that's why the vortex is created. You see the particles moving around them. It's better with the dark field. I don't know if I can do dark field with this optic. No, I can't really do dark field on this one. I have to change optics again. But if I defocus in here and there, you can see the particles dancing around still a little bit. There you go. There's the swirl from all the cilia action. So yeah, I find these guys everywhere. I find them in Baylands. I find them in ponds, even at the zoo. That duck pond at the zoo, they've got some great slides from. Really cool. Love these guys a lot. So let's move on to something else. What's next to it here? There is another flagellate. I can tell because I've seen this guy before. See, it has that kind of unusual shape. There's actually a curve here. It's kind of a curved structure right there. It's kind of nice oval margin, but then there's like an indentation there, and that's where the flagellum comes out. And I don't remember if these guys have 2 flagella or just 1. Right now it's not doing anything, so it might actually be kind of not doing so well, or it expired, or maybe just resting. Sometimes they just sit there for a while. But it's a flagellate, and they've got that certain shape. I'm zooming in just to, I'm focusing in and out just to see if I can see a flagellum moving. I can see a flagellum on the margin of the cell membrane, not doing anything, so it may just be resting. You can see the diatoms near it are like little submarines moving in and out. Those are single cell plants in a glass shell. It's actually a silica shell. And this is why diatomaceous earth used to clean your pool filter works so well, because it's abrasive, and also used as a filtration medium. And it's made of millions and millions and millions of little diatom shells. There's a whole cluster of different diatoms here. And you can see some spirilla bacteria here too. So those little wiggly guys going back and forth. Boy, I gotta zoom in on that some more, I think. If I can. Let me do a digital zoom real quick, just for easy peasy, but let me see if I can get a focus. See, there's some little, it's like a partial corkscrew zipping back and forth. They'll stop once in a while. You see it's like a bent piece of rice, because it's kind of weird, short, transparent things. It's a local bacteria called spirilla, and they're very quick swimmers. There you go. See a little zigzag going back and forth? It's really hard to slow them down. I'll have to do some slow-mo video one of these days with a dedicated camera for that, just because it shows it so much better. But you can see how much smaller they are than the diatoms and everything else. These are actually bacteria. There's some diatoms right there for scale, which are not massive diatoms. But yeah, this is a whole cluster of bacteria going in and out of this junk. And the corkscrew ones that are going in a straight line, left and right, that have this little bent shape to them, like a little partial corkscrew, those are spirilla. Really interesting little creatures. There you go. There's one's kind of stopping a little bit. And they also have flagella that go out in different directions and allow them to spin. And it's kind of funny to think, well, why would a corkscrew? What's the point? Because in bacteria you have cocci, which are the circular, like round shapes. You have the bacilli, which are rods, and you have the corkscrew ones, which are either called spiral, or like there's actually a specific type of organism called a spirochete, which are much longer corkscrews. And the corkscrew, actually, when you look at it, is very clever, because when they pretty much live around this muck, right? It's very thick, gelatinous, detritus muck. You know, plants sticking together, plants decomposing, gelatinous, you know, proteins breaking down, stuff like that. Well, if you're a big sphere, or even a small sphere, you're a blunt object. You're not going to penetrate very easily into a bunch of muck. It's going to take a lot more force. And if you're a rod, you also could be a little too blunt, and maybe can't quite get the mechanical advantage to get into the muck. But if you're a narrow little spiral, just like how your corkscrew goes into your wine cork very easily, they just twist and go right through the debris with the least amount of effort. It's very clever stuff. There might be other reasons too. People discover why they look that way, but the most likely thing I've read about and actually witnessed is the corkscrew allows them to go through all the muck, which is pretty clever, actually. So more diatoms moving around here. So I had to zoom in a little bit. It's not the sharpest, but I wanted to show you the spirilla, because they are pretty small. There's a couple right there. There was one wiggling around. See how they stop? It's like a little partial corkscrew, or like almost like a C shape, depending on when they stop. And you can see them go in different directions. And go, it's just like they're really fast. They have a very good acceleration, and they go all different directions. So yeah, bacteria aren't just these little wiggly things that just sit there. Some of them are actually really good swimmers. Go zipping through medium. There's one that stopped right there. So kick in again, start to wiggle and zip right off the screen. Oh, it's changing direction. That one's really sitting still for a while. Usually they don't sit still for very long unless they're not doing too well. See all the other ones around. Ooh, there's a big guy that went by. So yeah, that's some bacteria for you there. So if you haven't seen bacteria in a while or not for a long time, that's a really cool one. A little corkscrew bacteria called spirilla. All right. Zoom out a little bit because the digital zoom is killing the quality. Let's go look for some other stuff. So you've seen, again, these are the flagellates I was talking about. Make sure I get this centered because I've got a different view here than I do in my scope. I think that's pretty centered. A little bit off center. Now we're lined up. Okay. So these are, again, some other flagellates that just either out of it or taking a break or something. And you can see that notch at the top. If I defocus, you can really see that notch, and that's where the flagella, or flagellum, I think in this case, just one of them comes out. Little diatom up at the top there, going by. Again, like little submarines cruising around. There's a diatom right there. Funny to see all these different types of plants, single cell plants swimming around. Ooh, there goes that little Stylunkia dude. Yeah, it might actually be Euplodes. I think on second thought, I said it was Stylunkia. It might actually be Euplodes. Again, they're both some of my favorites. So it doesn't matter if it's one or the other technically, but I think I mislabeled it the first time. There's another little tiny flagellate, wiggling, wiggling, wiggling. Very low contrast, hard to see it here. I need to switch to dark field to show it better, but it's a kind of erratic movement. It's going in circles. You can see the flagellum making it kind of shake around like that. Not nice and smooth like a ciliate moves. Like a paramecium, very smooth operators. Okay, that guy's really fast. Again, but the movement, so definitely flagellate. Constantly in and out, up and down, very hard to focus on. There goes a spherical flagellate cruising by. You have this tumbling motion. Yeah, all kinds of diatoms here. Look at that. A cluster of diatoms. See if I can get a different viewpoint here. This one optic's a little picky compared to the other ones. So we're about it's about 100 power here. No, sorry. This is 400 power. 400 power. So these are pretty small diatoms. Another diatom. Lots of diatoms here. Not sure what that thing is. But there's a flagellate floating next to it. Let's go to the margins, see what's happening here. A couple diatoms together. All right, what we got here? some debris. Trying to get some air bubbles forming as the water is evaporating from the slide. And a diatom there. Big diatom there. Let's go all the way to the edge of this slide. Starting to get crystallization there along the edge of the slide. It is salt water, so you start getting some weird crystals. Not just sodium chloride, but other things. Of course, table salt is perfect little cubes of salt. It forms a nice little cube when you look at your table salt under the microscope. I think I've done that before. I should do that again. But salt can crystallize in this format in different ways, kind of like a little crust. Yeah, I had some stuff on the margins earlier, but I'm not seeing them. There's some nice blocky crystallization there, very sharp. Pretty cool. What else we got? More sharp. Look at that stuff. Starting to look like Fortress of Solitude from Superman. Pretty cool. Look at those guys. Very sharp. These would be great with polarized light. I'm going to get a polarizer rig set up. I've seen that X pattern before. That's pretty cool. Focusing through, showing the thickness and the light refraction as you change the focus. Gives it a 3D look. All kinds of junk on the margins here. Let's go back to a lower power. Let's go back to 100 power. Kill some of this. I should just go back to dark field on this one. I think we can do that. Maybe do some oblique lighting here. That gives you some cool 3D looks. Almost a polarized effect. All kinds of crystals there. All right, let's go find some more critters if the slide hasn't dried out yet. I think it has actually. No, still good. There you go. A little more of a dark field approach here. See some more details. Dark field's great because you can see all the particles, like all the little white dots just pop out. So whether it's debris or a bacterium or whatever, you see all that little tiny stuff there moving, like twinkling stars. Fields of diatoms. Back to the debris I started with. Clusters of junk. That's most of what's on this slide, so not bad for a first slide. Let's switch it out. Let's see here. I think the music actually stopped. I thought the music was going to be continuous. I guess it only played the one file and stopped. Give me a minute here. I'm going to fix that. That's not the way it's supposed to go. I didn't notice because I had it on mute. Because the way I have it set up now, if I have the music going and I'm talking, I can hear both repeating in my ear. Let me fix that real quick. So I'm going to kill that background music section. And that's not what we want. Let's do this real quick. I don't want to waste too much time setting this up, but it does help the stream go a little nicer when there's some music in the background. Media source, is that what it is? No. OBS is still funky. Always trying to figure out some new tricks, and I think it's this one. No, that's not it. Where is it? There it is. I think this will work. Let's try dumping the... See if this works. There we go. I think that's working. Where that tells, let me go back to my test stream on my phone. Seems to be working okay. Okay, I think that's good enough. Go back to the stream here. Okay. Sorry for that delay. Just want to make sure there's a little background stuff. Let's see where we are here. Oh yeah, I was gonna make another slide real quick. Let's just do. I'm not gonna do a crazy long stream tonight, but haven't done one for a while, so I wanted to get something done. All right, let's prep this. Bear with me making another slide. Mix up my water sample a bit, see what else we can get out of here. This one's got a little more debris on it. Might be more interesting. Okay. All the same stuff. I'm just trying to look for some new things here. I just gotta check these little debris piles. Those are fun, usually. Got some little guys diving in and out of that stuff. There's actually a nematode in there. Nematodes love junk. See all the movement inside there? And you can see it's peeking out on the right side just a little bit. So nematode action. The slide's shaking a little bit more. I put too much water on the slide, so every little bump I hit on the table makes the whole thing shake. This is the song of the nematode here. Peeking out. It's going a little bit closer. I'll lose the dark field, but check this out. So you get a better view of the body of the nematode here. See, it's in like a tube. It's like a hollowed out stem of something, and it's hanging out inside of that. Little diatom on the top, nematode in the tube, peeking in and out. Good thing these things are mostly transparent, otherwise I wouldn't be able to see this guy at all. All this debris. So, you've seen me cover nematodes many times before, so I'm not going to go into too many details on them, but they're very interesting little microscopic worms. Found all kinds of environments. There are even some the ones that they have a pretty tough cuticle on the outside. They can live in some harsh environments. There's even ones called vinegar eels that can actually live in acetic acid. We had them in lab once a long time ago. Let's do claustrophobic here. Let's go back out. And readjust this. You can see him peeking out a little bit there. So that's fun. Some more worm action. Actually, you know what? That's way too flexible to be a nematode. I take that back. That's one of the that's one of those really cool oh damn, that's a yeah, I've seen these guys before. It's a really long ciliate. Moves a lot like a nematode, but has a lot more control instead of being whipping around like that. Damn, I have to look that up again. I forgot which one that is. You can see how it stretches out that top part, almost like a neck. Probing, probing, probing. Oh, no, it's leaving. No, don't leave. So I can compress, and then it can thin itself out and bend part of it around like it's actually like a neck with a head part. It has a dominant front part. Damn it. I lost him. Where'd he go? Where'd he go? Where'd he go? Yeah, I've seen him before. There's a really long one called Lacrimaria that does a really long extension of its cell membrane into this long, like a proboscis kind of thing. But that one's different. It's not Lacrimaria, but I've caught it before on my streams. Don't know where it went. Let me go do another search here. Extra water is really making it wiggle too much. Actually pretty annoying. I have a little micro pipette thingy that I need to start using. Where you actually dial in how many microliters you want to insert. It goes from 10 microliters to 100 microliters. And can dispense a perfect drop every time. That's one of those flagellates I found earlier. Remember the ones that had the little depression at the top? That's what they look like when they're not sitting still. Let me do a quick digital zoom to cheat. Just so you can see them tumbling. You can see the flagellum behind them, wiggling around a little bit. And they have that little notch at the top where the flagellum comes out. And you can see the flagellum wisping around a little bit. Not a great image, but you can at least see it. Yeah, zoom back out again. Let's go back to a little bit of bright field, and you can get a look at the color of them a little bit. So flagellum generally shows up better in dark field, but you get the idea. But see, they just like tumbling behavior. Let me zoom back out and try a different optic. There's a diatom. There it is. See it tumbling, it's got some color to it there. There's those little smiley diatoms. It's like a little smile upside down. So here's this little tumbling flagellate. I don't know this one, but I've seen them around. I have to look it up. But it's cool when they tumble, you can really see the 3D structure. It's hard to smoothly do a XYZ at this magnification, but it is doable. It's just a little stiff. So, get an idea what those guys look like at least. Pretty cool. Let me zoom back out again. Anything else stand out here? All the same stuff. Nothing eye-catching yet. Trying to find some bigger, more exciting stuff. I mean, these guys are fun and all, but need some showpiece critters. Let's do one more. I'm gonna do one more from that sample. Stir it up a little bit more. I'll get to some other items. Alright, let's see if this dug up anything else interesting. No? Wow. Wow. That's why. I was looking in the wrong area. Now there's a cover slip. Again, tons of those little flagellates there. Cool shape, but very small. Nothing in the clusters. Oh, sorry, I have the brightness too high on this thing. There we go. Cool. We should clean that slide better. Got some junk on it. Yeah, that wasn't good. Let's do something from the other pouch here. So, down the walkway a little bit, I went to this little section here. Let's see, There it is. So into this little pool down the levee. So there you can see the actual levee. There's the levee you walk down. Curve goes down quarter mile, kind of curves around a little bit, goes by the airport. So there's a mud flat on one side and lots of pickleweed, cordgrass, and other plants on the sides like gum plant and stuff. And then there's little pools everywhere, and the pools have lots of little algae and beautiful, colorful things growing toward the bottom. This is kind of a reddish purple color. It's very, very nice, almost pink. Picture doesn't quite do it justice, but it was very, very pretty. And it's too deep for me to get a sample of that. It's way under the water, but I got some samples of these plants here. Some algae growing in the water there. And also further up, I got some mud from these mud flats near the entrance. And you can see there's some kind of greenish tinge to some of the mud here. There's some algae growing on top of the mud. And if you dig in just a little bit, it's a lot of sulfur bacteria out there breaking down sulfur bonds. So it's very stinky. But not stinky as in rotten, like it's not from like garbage, kind of nasty stink. It's just kind of the rotten egg smell, but not super strong. But when you start breaking through the mud, you get the smell of that wafts up, and you go, "Ooh, that's really strong." So the sulfur bacteria in here, and so the next couple samples are those things from the pool, those algae from the pool, and the mud. That's there. So I'm going to switch briefly to the dissecting scope. Okay. I'm going to take a little forceps and I'm going to grab some of that algae out of the bag. There's water with it, but it's been in the fridge for a whole day, so probably not a lot of stuff swimming in it. And I have mud in this another corner of the bag, so they're not completely isolated, but let's zoom way out and focus. I have to play with some different lights I have here to get a different result. So there you go. You can tease out the little hair-like algae. What I can do is I can grab a piece of it and put it on the other microscope for a close-up examination. This only goes to like 60 or 80 power or something like that. So if I zoom in a bit, you'll see what I'm talking about. Start to see through it a little bit, but then I need some backlighting. So I'm doing top light now, I'm doing backlight. Like going through. Sorry, now I'm doing some backlight. Like you'd have on the other scope. All the top lights are off. basically have to adjust the light so it doesn't overexpose. So you can see some of the structures there. But again, you're not getting a lot of detail because it's too far out. So I have to break a piece of this off. Tease a little bit of it off. Put it on a slide. I'm going to go ahead and mix it with the water from the other site. I mean, it's pretty similar to water, even though it's grabbed from a different site. It doesn't really matter at this point, but I need to hydrate the fibers a bit so they spread a little bit better. Try that, they're a little clumped. So I'm going to grab these fibers. Whoa. I'm going to take the whole thing out. And I'm going to take a little bit of water out of the other sample that I had. Just so it's happy. You can imagine why I don't put tap water on it. Because this is supposed to be salt water, or at least brackish water. And it would not be pretty playing with the osmotic pressure like that. So I'm going to switch back to the other scope real quick. Cut down the light. So now you can see. Oh, got it done. That's actually a nematode this time. Last time was a false alarm. That's a nematode. It's probably clinging to the algae. But you can see that. Start seeing the cell structure there. I'll go in under the level. Get individual cells there on the plant. Yeah, nematode stealing this photobombing. I got a nematode photobombing on the algae in the background there. Let me zoom in a bit more. Give it some more light. And you can see the cell structure. Oh, there's a diatom photobombing this time. Right on top. Something else right on top as well. So there's the cells there, and you can see chloroplasts in there. So those little green dots inside the cells, kind of hugging the perimeter of the cell membrane. So actually chloroplasts. I don't want to go full don't want to go full zoom with the other optics. Let me do a digital zoom here. You can see a little better. Focus a bit there. And see a little All those little guys there, little chloroplasts around the rim. So that's pretty cool. And they're not like, you know, perfectly orderly square or round cells. They're all kind of, you know, irregularly shaped cells all connected there. And if I went a little bit further, you'd probably see a little more, actually see some cell activity with stuff moving inside each cell. Some plants that, some water plants are interesting. They have this phenomenon of, it's called cytoplasmic streaming. It kind of keeps the chloroplast moving around in kind of a motion like that. I think I've seen it counterclockwise and clockwise. I forgot. It's been a long time since I've seen it, but you can actually see the chloroplast moving around in the cell. And I believe, if I remember correctly, it's just to guarantee you have a good light angle hitting your chloroplast at all times. So if you're just sitting, the plant isn't moving much, then the chloroplast buried in other chloroplasts aren't getting hit enough. So by streaming them around, they're all getting exposed a certain amount to maintain efficiency. Get as many chloroplasts firing, making photosynthesis work as much as you possibly can. So yeah. And I forgot what species of algae this is. I'm gonna have to look this up, but there's a smack of it. There's just a ton of it. A ton of it, as you saw in that picture. I'll refresh your memory if you didn't. Not that. No, not that. No. Not that. It's the other shot. This spot. So all that green in the water there is just tons and tons and tons of this stuff. That pool right there, if you go from left to right, kind of diagonally, that's probably about 12 to 15 feet, maybe a little more than that. So that's a lot of these little plants. So that's what we're looking at here. Okay, let's switch to, let's start looking at that mud a little bit. That was kind of cool. You usually find really cool stuff in the mud. Okay. I have to use my other eyedropper for this one. Now it's tricky with the mud because it's very dark mud. So it's easy to get so much on here that you won't see anything. So another trick is to dilute it. So I'm going to have to go back to that other sample bag. I didn't have any tubes with me, so I used some really nice little good quality Ziploc bags to get my samples. And they travel well, they just squash in your pocket, so you need them. So I'm just going to drop a little bit of water on here so I can stir it up. Because right now it's very dark and clumpy. And it's going to be really easy to screw up the opacity. I won't be able to see anything in here. It's still pretty dark, but let's see what we got. Always have to clean these slides and cover slips. They come clean, but they're all dusty. So you always have to wipe them down. It's kind of a pain in the butt. All right, here we go. Let's see what we can find. I already got some stuff in here. Look at that. Almost always find some like worm kind of things. Get some nematodes, some ciliates. There's nematode right there. Speak of the devil. Nematodes love that mud. Let's see what else we got here. Lots of those same. Whole lots of diatoms here. Actually, no, that's more. Those are blue-green algae there. Those long rods, are blue-green algae, also called cyanobacteria. But they're not fat enough to be a Silatoria. Let's see what they are. There's a decent one there. Getting pushed by some diatoms. Like it's a tugboat. Diatoms are pretty determined. They have a lot of oomph pushing stuff around. Okay, I think I have to adjust this again. This is out of whack. There we go. Okay. So it's diatoms and cyanobacteria. Let's zoom in a little more. Give it some light. See, the diatom is pushing all that. It's like tugboats. Because the cover slip is making the z-axis of the area very compressed. So they're big enough they're actually running into that. Well, they're actually going underneath it a little bit, but they're running into it still, pushing and pulling it. So that's all diatom movement there, moving these giant rods of blue-green algae. It's funny. So you can see the segments of the blue-green algae there. If I go to the open section there, see there's some markings on it. It's got segmentation there. It's like a colonial. It's not an algae. It's a blue-green algae, so it's more related to bacteria. But they're photosynthetic, very primitive, but everywhere. And they can make really large structures. You can see here. Look how big that diatom is, and look how big that rod is. The cyanobacteria rod. It's not. Yeah, it's not big enough to be a Silatoria unless it's a small one. But it's similar. Now the Silatoria is moving, see? So it's moving and the diatoms are also moving. So it's all these like plants and plant-like things that have locomotion. All kinds of fun stuff at this scale. A little flagellate bouncing around there. So yeah, pretty cool stuff. Let's go out another level. See what other thingies are around here. So a lot of blue-green algae, a lot of diatoms, a few nematodes. Another nematode there. And the junk. They love the junk. I have one worm video I shot probably a year ago in this kind of muck. I keep forgetting to edit it and post it. It was one of my local videos I shot. It wasn't a live stream. And it's a beautiful, huge worm. And it's kind of a dramatic sequence because there's a lot more particles that time than this. It was kind of like a rocky mud, but tiny, tiny microscopic rocks. And a bunch of it was navigating through the rocks just fine. And then the water started to dry up on the slide. And as you've seen on my streams many times before, there's the wavefront of the water-air line coming in. It's like the Terminator coming in, and normally they just kind of go off in a little pocket and they sit there for a while and swim around. But so there's one of the air pockets there. If you watch it, it's slowly growing because of evaporation, which eventually dries up the whole slide. So this was advancing, but much quicker. And there's a bunch of these rocks and stuff, and the worm was caught in the debris. And normally they kind of feel it coming or whatever, and they kind of move out of the way and have freedom to go somewhere else. But this one was not that agile or whatever, and it just kind of sat there. And the wavefronts came in from different sides and kind of trapped it. But it pushed it a bunch against some rocks. So it's like the unstoppable force versus the immovable object. And this poor soft-bodied guy got stuck in the middle. And normally it could just squeeze right out, but the pressure was immense. You could see this big wavefront was coming, and the rocks are very tough. So it actually got pinched, and it was kind of wiggling itself up and down trying to get out. And it pulled so hard, it looked like it was going to break its own cell membrane. Because remember, it's a single cell. So did I say worm? It's actually a single cell. It's actually a ciliate. It wasn't a worm. It was a giant ciliate, which I forgot which kind it was. And so here it is, a single cell acting like its little neck is being squeezed, and the top part is moving around like a head, like trying to find a way out. The body's trying to pull back the other way. The bulk of the body is pulling down, and it started twisting on that little junction trapped in the between the rock and the thing. It was very dramatic. I'm like, man, this poor little thing, and it's twisting and pulling. It looks like it's going to snap, and you see it twist, and the contents of are just kind of sloshing about inside, and it was super, super dramatic. And I tried adding water to the slide to give it some more room to move. It didn't quite work. So finally I took pity on it, and I took the cover slip off and rinsed it into the container. I couldn't find it again, so I don't know what happened to it, but yeah, it's just all this drama with this poor single-celled creature that looked like it was like an animal that has his foot caught in a trap or something. It's very, very sad. People don't usually feel sorry for single-celled animals, but it was very dramatic. So I have the footage somewhere. I have to find that just to show the drama. I didn't obviously I'd leave it too long because it's kind of like torture at some point. I felt bad for the thing, but so yeah, that's what happened. It's very interesting stuff on these slides. But it was a similar field to this, with lots of debris and rocky stuff and nematodes and you see this area already dried up. Everything caught in this matrix is all dry, all the blue-green algae. There's no water there. The water's moved on down here. So yeah, not much in the mud. I might save the sample and do it another day or so, because I'm kind of getting tired. I was gonna try and do a much longer stream tonight, but I'm kind of out of it. But I didn't want to wait. I fell asleep last night early. Be right back. I have to check something real quick. I'm muting. So a few more shots. This is the area. It was, like I said, there's the bay in the distance there. and all these various pools on the right side of that levee. And this is all mostly pickleweed out here. Pickleweed, some cordgrass, and a few other plants, but mostly pickleweed and cordgrass. It's a really dominant plant out there, or series of plants, but they have these stratifications, little zones, where cordgrass is like way out by the water, and you can see the lighter green color in the distance is more cordgrass sticking above the pickleweed, but pickleweed has this kind of a rougher look to it from a distance. And the little branches do look like little pickles. It's kind of funny. But they both are very good at adapted. They have adaptations for living in salt water, taking in the salt water, and immediately pumping the salt water either to tissue to basically fall off as the pickleweed stores the salt that turns red, and the pickles fall off. As cordgrass has a mechanism to actually pump the salt outside the tissue into salt crystals on the outside of the leaves. I'll show that here in a second. Here you can see there's a kind of a boardwalk out here, and more of the cordgrass in the distance. And again, the bay is right there, and goes all the way down off camera to the right, down to this what's called the South Bay, which is where we are here in this picture. And across the bay is the East Bay. So it'll be like Fremont over there, and Union City, stuff like that. So anyway, so let's go to another shot here. Near the trail's edge, you get this salt grass, this kind of tufted stuff. It's also adapted to salt water, but not as handy as pickleweed and salt and cordgrass at handling the salts. You also have this yellow flower called gum plant, genus Grandelia. So it makes this kind of gummy substance to keep it from drying out, and also to protect it from insects and stuff. And here's the salt crystals pumped on the outside of the cordgrass. I actually picked this so I could get a better shot in the light of the glinting salt crystals on the outside of the leaves. So very cool adaptation. I'll try to show you on the microscope here, but it's been in a bag for a day. The salt crystals are kind of wet, because the salt is hygroscopic and pulls water out of the atmosphere, and attracts water basically, so it doesn't stay dry like this for long if the air is moist. Was I not showing that? Oh man, sorry. Thought I was showing those damn pictures that I was talking about. I had the wrong image up, the wrong page up. Let me go back through those again. That's where the sample was. That's the cordgrass salt pumped on the outside. Little crystals there. It's really cool how they do that. That's the salt grass, the tufted stuff. Not quite in the salt water, but it's touching the salt water. It's not as durable as pickleweed and cordgrass are. And there's the gum plant, the yellow flowers. This is the part of the cordgrass I was showing the stratification, where it's pickleweed in the foreground, cordgrass more in the outer areas. And the bay is actually right there behind the boardwalk. And the hills and the shoreline in the distance is part of the East Bay. Actually, Leslie Salt has a plant out there where they actually extract salt for table salt. Big salt piles out there. You can't really see them from here, but I'll take some pictures someday, show it to you. You can actually see them across the bay. And then then this is another shot showing mostly pickleweed around here, with some cordgrass mixed in and out. Another big boardwalk going out here, and the actual bay there. San Francisco is that direction, and San Jose is that direction. And again, that's the East Bay over there. So it's a really cool place and easy to drive to from most of the Bay Area. So that's the tour of what we're looking at. And this is what the cordgrass looks like now. It's a little wet, but you can still see it's a bit shiny where the salt crystals were. I started collecting water drops because, again, salt is hygroscopic, pulls water out of the air. But that's why the cordgrass is still wet, because all the salt was on there trapped in the water that was in the bag. The humidity. It's a big specimen, so it's hard to show much more with the dissecting scope. So I'll go through a few more specimens here, and then I'll call it quits because I'm a bit tired. Let's see, what's this? This is the salt grass. See, it's also got salt on it as well. Same adaptation as cordgrass. Now it's a little more moisture than salt because it's the salt attracts so much moisture. But you can see by the shape, I'll zoom out a bit more. It's got that more tufted appearance to it, kind of scraggly tufted appearance than the cordgrass has, which has bigger leaves. And then I've got some gum plant. The flower got a little smushed, but that's the gum plant. And also it's wet because the bag has all that salt in it. And I'm not seeing, you can see some of the, yeah, this one doesn't have a lot of the white sap on it. You normally find outside the gum plant it actually secretes a lot of white sap. And occasionally I find some cool insects on the leaves, but not now. And what else we got here? Here's a nice shot of the pickleweed. You can see the, looks like little pickles. And as they turn red, they're actually storing the salt. And when it gets too much salt, that part dies and falls off. So that's their adaptation. Instead of pumping the salt outside the plant, it just sends it to a segment of the plant, turns red, and drops off. So normally it's that nice green color. So a bunch of this is red. Has a bunch of red tips ready to die shortly here and fall off. And the little salt marsh harvest mouse actually eats this. I saw it eating a little piece of one like corn on the cob once. So some animals do eat the pickleweed for sure. Alright, well that's all the plants I had. I went through most of the specimens already, so I'm going to call it here. A little tired, so thanks for joining now or later if you choose to watch it later. Sorry, the stream's a bit rusty again. I haven't done it for over 6 months. I had to reset a bunch of stuff up and get my groove back, so not as efficient as I usually am. Usually I can go 3-4 hours on these with all my stuff all set up, ready to go, and not so many tech issues, but hey, it was fun to do them again. And you get to listen to some really nice JX3D music. Go look up JX3D, exactly how it sounds, on Bandcamp. buy a bunch of his albums. They're very fairly priced, really good music. And I'm going to end it there. So thanks again, everybody. And I will see you soon with some more critters. And take it easy.